All right, well, good afternoon, everyone. I'm here to, again, talk to you about multi-organ platforms and how we can utilize them. And mostly this talk will be about the development of the platform and some validation. So I don't need to really explain why we use organs on a chip at all, but I'm just going to put it into the context of what we do in our lab, and that's really not to build a platform or technology or organ on a chip that's one size fits all, but really to make them fit for purpose, and so only designing each platform or each organ on a chip with the end goal in mind. So what disease are we trying to study? What's a biological mechanism? And only engineer the complexity needed to answer that specific question. So that we don't need to, have everything in a chip. It just gets too complicated. And then you lose your robust reproducibility. But just really simplifying what's the question I need to answer and what are the inputs that will get me there. And so for this study, our goal is to develop a patient on a chip model of a systemic disease. And that systemic disease is doxorubicin-induced cardiomyopathy. So our design constraints were that it be patient-specific, so using a patient's cells, so all their iPS cells to make that specific patient in a petri dish. Have these tissues have high biological fidelity so that they're actually predictive. And this really comes into play when you're using iPS cells. And then connect these tissues and connect them in a way so they're stable for weeks to months, so you can actually get systemic disease, and then being able to get information out of the system, because if you have a perfect system but you're not able to get these readouts, it's a missed opportunity. So having functional longitudinal readouts, and then having this platform that's plug and play, so that way I can use it to answer this question, but you can use it to answer another question, and we're really not sacrificing any science because of our platform, but really keeping the platform amenable to whatever biological question you want to answer. And then, of course, add on that complexity as needed. We'll have immune cells you'll see later on, but we don't have things like innervation, or we're not looking at scaling. But there really is a big need to leave room for everything we're not including so that we can continue to develop these models. So just a quick case study, because we're using patient-specific, why tissue maturation is important, because you'll hear me talk about maturation throughout the whole talk. And so because we're doing patient iPS cells in a petri dish, they're really plastic and naive, and so we work a lot to coax them to become more mature, and then we need to keep that biological fidelity, because if they're not predictive, it ruins the whole point. So iPS-derived cardiomyocytes are a really true case in point. So if I have adult patient cardiomyocytes and I expose them to isoproterenol at increasing doses, I see in the green, I have this increase in force and a corresponding increase in calcium. But the second I take iPS cells and differentiate them to cardiac cells, they may beat, they may look very nice, but I can do that same exact response, and I'm not seeing the increase in force or calcium. So a couple years ago, we came up with this methodology to take iPS-derived cardiomyocytes, and at a very early stage, so as soon as they commit to that cardiac lineage, we mature them. So we put them in a 3D hydrogel around these pillars, and we make them work out every day with electromechanical stimulation, and this really gets them to be mature. And so now we can do that same assay. I've got my beating heart tissue, I've intensity trained it with this ramped regimen, I expose it to isoproterenol, and now I see that expected physiological response. And so it's really important to, within each individual tissue that we're engineering, make it physiologically predictive and have a set of reference compounds to, as quality control to ensure that. So now the strategy comes into play where we've worked to get this biological fidelity because we're using iPS cells, but now we want to connect all these organs together to have a multi-organ model without sacrificing it. So there's this balance of, how do we connect organs with very different media requirements and cytokines and growth factors, but keep them as their original phenotype. So we're always trying to balance that. And so our strategy is to kind of take a page out of the book of our own body. And so in our body, we have our organs kind of in their own niches, and then they all talk to each other through vasculature. And so we've greatly simplified this by just unrolling the vasculature and putting it on a transwell endothelial insert at the bottom of each well. So you're flowing this common vascular media with circulating immune cells below each tissue, and then you have the tissue niche above. And so what that looks like in a platform is this really modular open setting. It's polystyrene, so it really doesn't absorb drugs. As you heard before, we drop those transwells in, you coat them with endothelial cells, and then you can plug and play, any engineered organ you made in the lab, an organoid, a biopsy, a monolayer. It's really open and amenable for your studies. And then you're flowing cells underneath, so immune cells and your endothelial media. And then you'll drop your tissues above. So you have tissue-specific media, and we leave everything open so it's really easy to sample over time, and we don't have to worry about bubbles. So having an open reservoir acts as a built-in bubble trap. So we've made the platform, we have our transwells, but first we need to engineer our tissues and then transfer them to the platform. So I'll kind of go over that briefly. So the heart I already went over, so we take cardiomyocytes and supporting cells, fibroblasts. You'll notice every tissue has a main cell and then a supporting stromal cell, and then as the years go on, we'll add more and more complexity with other cell types. But we make cardiac tissues, expose them to this increased regimen of electromechanical stimulation. They show increased in the maximum capture rate, so how fast they can beat, and how fast they can beat at a lower voltage that we're applying, so that excitation threshold, and then responding to calcium. So as we add increasing doses of calcium, they increase in force, so it's a nice physiological response. In the liver, we take hepatocytes and fibroblasts in a 1-to-1 ratio, make them into these spheroids by just spinning them down in your common agar wall plate, and then we put them in a bigger community by just encapsulating them in a larger fibrin gel. And this 3D microstructure lets them really continue to secrete albumin and stay stable over time. Our bone, we actually take actual bone from bovine scaffolds, we'll mill it into our desired shape, and then we decellularize it so all the bovine components are out, leaving just the ECM, and then we'll re-put in MSCs, which become osteoclasts, so your bone forming cells, and then monocytes, which become osteoclasts, your bone resorbing cells. And you'll see we have things like the osteoblast leading edge. You can look at bone density over time and actually see remodeling. So it's a very active remodeling bone that we have in the system. And then our skin is made in the Angela Cristiano lab, but basically using your common transwell assay, you'll take collagen and dermal cells. They'll form a nice dermis, and then you put your keratinocytes on top. As soon as you expose them to that air-liquid interface, they stratify and become epithelium. That's quite intact, has great barrier function. So then for our vasculature, we put endothelial cells and then MSCs just as a supporting stromal cells, and we put them on a membrane, and then we expose them to ramped shear stress. So you don't want to, just start flowing right away, because then the cells will come off. So we start pretty low, and then we ramp up to about 1.88 dynes per centimeter square. We use a 20 micron pore size, and we picked this because it was the optimal kind of Goldilocks zone where we had minimal artificial membrane and more cell-cell contact. But 40 microns was just a little too big, and you were getting holes. So we want, a pure endothelium with as little plastic or transwell in the way. And you'll see that kind of is replicated in the tear at these different pore sizes and then shear stresses. So to validate the platform, we wanted to look into a couple of things. So mostly for systemic disease, because it's a multi-organ platform. So first off, are our organs that we've put into our system, do they actually talk to each other? Do we have inter-organ communication? Can you keep them in culture for the long term without sacrificing the individual tissues? Looking at individual responses to injury. So we want to look at systemic disease, but also local injury. And then just showing its utility. So for PK/PD modeling and predicting this doxorubicin-induced cardiotoxicity benchmarked against clinical results. So step one, do our organs talk to each other? And so we did a lot of cytokine assays where we're looking at, this cell secretes this, does it go into this component? But because we have so many stromal cells, there wasn't like a true marker that we could really follow, so we decided to engineer the heart with an exosome reporter. So it's labeled CD63 in cardiomyocytes are labeled with GFP, so any exosome, extracellular vesicle that the heart is secreting will have that GFP label, and we can track where it goes in the system. You can see this on flow. Our cardiomyocytes have the GFP marker. It's up to, you can see the CD63 in the vasculature, and then we can look at it in other organs. So while it's primarily in the heart, because we've engineered it that way, it is going to the other organs, and we're seeing that they are communicating with one another. So check one, we prove that they have inter-organ communication. Now let's look at inter-organ communication for the long term. And so we looked at the tissues over 4 weeks. So the inter-organ condition will be the one I've just described, so having a vasculature barrier and then having all your organs. We have this mixed media approach, which is just an empty barrier. So everything's the same, the media amounts are the same. Basically in the top of each chamber, you'll have each tissue's specific media, and then circulating you have the endothelial media. So the same in the mixed chamber, and then isolated, we have no connections at all. It's just more similar to a standard well plate. So looking at it over 4 weeks, we see that the biological fidelity is pretty much preserved in the interorgan and isolated group. So we've got those cardiac striations in the heart, in the liver, you've got albumin. We're losing this in the mixed condition, so you're losing your cardiac striations. In the liver, we use albumin. We get calcification in the liver, ossification in the bone. We're losing our epidermis in the skin. So that mixed media condition isn't supporting each individual tissue niche, but by having endothelial barriers to separate them out, we're able to keep each niche functional at least over 4 weeks. So this is a vascular endothelium. You'll see the liver becomes a little leakier, which we suspect may be acquiring more of a liver endothelial phenotype. It's represented also in the tear, so it is staying stable. And then looking at immune cytokines, so the first panel, the first left are the interorgan over day 2, 12, and 28. They stay pretty consistent, whereas you're really losing your cytokine secretion and signals in your other ones. So having the vascular endothelium and a really stable environment is keeping your signals stable over time. Looking at tissue function, so you also have preservation of your cardiac tissue contractility, albumin production in the bone, only in the interorgan group. So when you have the vascular endothelium and tissues talking to each other, do you have trap activity? So showing that your osteoclasts are still functional. And then in the skin, you still have that maintenance of the epidermis, but you're losing it in the mixed condition. We did global proteomics of the tissues, and we looked into the shared pathways. So what's expressed in all the tissues that's shared? And these are pretty different tissues, so there shouldn't be that much gene expression-wise that's shared between them. And so doing gene ontology analysis, it's pretty common pathways in the interorgan and isolated. So isolated, you see like metabolic pathways. Interorgan, you see like cell signaling, cell motility. But in the mixed condition, every single tissue shows markers for vascular development, skin morphogenesis, cardiovascular development, bone ossification. So I don't want to see bone ossification in the heart or liver. So this mixed media condition isn't really supporting these tissue phenotypes over the long term. We also compared it to the human proteome atlas. So these are adult tissues. We looked at the shared gene expression, just of the genes that are expressed in adults, what are also expressed in our tissues. Looked at the overlap. One caveat is that bone is bone marrow here because it's a rare tissue sample. So we see that there is an overlap, and when we look into how this overlap is correlated, so we tertiled it. If it's highly expressed in the adult, is it also highly expressed in our engineered tissues? And we see that the interorgan gives you the best preservation of this matched correlation. And when you zoom in even further, so these are the top proteins expressed in each tissue, so tissue-specific proteins, you'll see that the interorgan really closely matches that adult phenotype, whereas a mixed condition, you're just completely losing it. So what's going on in the mixed condition, looking at the cardiac tissues, we're seeing an upregulation of neural development pathways, conical development, things from other media like keratinocytes, so where it's becoming much more plastic and dedifferentiating. And this is expressed in our gene ontology for cellular components of the heart. So we've completely lost everything related to all the contractile units, so your I-band, sarcomere, intercalated disc, T-tubules, things that you really want for physiological modeling. Now looking at the specificity of systemic immune injury responses. So we took the heart and we cryo-injured it, and then we tracked where immune cells in the circulation went. Did they go to that site of injury, or did they just go everywhere? So only in the interorgan platform do we see that these immune cells only went to the site of injury in the heart, whereas in the mixed, it's not as directed, kind of goes everywhere. You can also see that they go to the vascular barrier just underneath. And then if we look at cardiac troponin secretion, we see a strong cardiac response, but it's pretty diluted out in the mixed condition. And we can carry this out for longer periods of time. And then we also just did a healthy heart next to an injured heart to make sure this wasn't a cardiac-specific phenomena and that immune cells were just attracted to the heart, and it's not. They're just attracted to the site of injury. As you can see, healthy, injured right next to each other, they're going to that injured heart. So looking at for physiological PK/PD, I'm not going to go into this too much, but just looking into experimental setup, we looked at using 4 tissues in the reactor, this mixed versus integrated or 1 tissue. And then if you go tomorrow to session 7, you'll hear Diogo go all into detail about the model we developed. We see the predicted responses, and our PK matches the experimental data. But when we really go into looking at the clinical relevance, and looking at microRNA biomarkers of DoxTox, we see that only the interorgan group shows miR1, which is kind of the only agreed upon biomarker of doxorubicin toxicity. So there's miR1 and then the dose of doxorubicin that you're getting. So the dose that we use is that dose, so we can kind of ensure the onset of DoxTox. And so we see that only in the interorgan platform, whether you have 4 tissues, just the heart tissue, or you're looking at the blood from the 4-tissue perfusate, do you see this miR1 biomarker. We took it a bit further at looking at cardiomyopathy, so heart failure after the onset of cardiodoxtox, and we compared our microRNA signatures using gene set enrichment analysis to a pediatric clinical trial, and we see in the 4-tissue and the 1-tissue that it's much more closer to what you saw clinically, so it's a bit more reliable. We'll do PCA clustering, and you'll see, so you've got clinical in the left over there, and then surrounding it, we've got all the interorgan groups, and then in the periphery, you see the mixed condition. So our interorgan 4-tissue perfusate, the blood basically from our 4-tissue is most matching or clustering with the clinical samples. And then if you pull out specific microRNAs of relevance for the onset of cardiomyopathy, it's much more specific when you're using this interorgan platform. So in summary, having mature tissues really facilitates your ability to do predictive drug screening or clinical modeling in a petri dish, but as you move to systemic diseases, you need to connect them in a way that preserves that biological fidelity. And then in the future, we're trying to scale this and make this more accessible. So instead of this reactor we make in the lab, we have funding from BARDA to make it into a multi-well plate, so the pumps are all on board with this really open format that's suitable for robotics, or in academia we don't have robotics, we have grad students and postdocs, so also our hands just doing that, but in a bit more high throughput that still gives you access to high content imaging, and it's very open, so you can really plug and play your tissues of interest but still connect them. So thank you for listening to me, and I'd like to open it for questions.